So we are Nervous System. We're an experimental design studio, and we started in 2007 sort of as a creative outlet for interdisciplinary experiments that didn't really have a home in our academic environment.
My background is in biology and architecture, and Jesse's background is in math and computer science, and we wanted to create a design studio where we could seamlessly mash up techniques from these very different disciplines.
So Nervous System is sort of this strange mashup of design studio, research lab, artist duo, lifestyle brand, software consultancy. We primarily, as a business, make and sell objects, sort of affordable everyday items like jewelry, housewares, ceramics, furniture, jigsaw puzzles.
Everything that we create is generated by software that we write. And in our studio, our practice is very centered around thinking about how to use computation to change the way that we design things. What you're looking at now is software that we created called Cell Cycle in 2009. And in our sort of app development, we really look at how to create new types of playful design experiences that try to really leverage simulation and what the web can do to make it possible for anyone to create objects. In this app, you can sort of customize in real time cellular rings, bracelets, and sculpture objects in a sort of physics simulation. And then you can order those things from us, and we'll print them in a variety of materials from plastics to precious metals.
So our work is often inspired by the complex patterns and forms that you see in nature, and not just biological things, but chemical things, geological processes, physical processes. And one of the things that is really fascinating is how this diverse set of phenomena, which sort of is caused by all sorts of different things, often has a similar underlying structure to it. And we can learn from those processes and sort of abstract them into tools.
So what we do in our studio is create generative software which takes inspiration from nature's process-based designs, and we focus on developing interactive processes that we can engage with. So instead of creating single static objects, we create dynamic systems, and instead of drawing structures, we're really interested in growing them.
So we're going to talk about a few different projects that we've worked on over the years that kind of covered different aspects of our work. So we're interested in how computation can enable customization and engage the user in the design experience. We're interested in how new digital tools can sort of reinvigorate traditional crafts. And we're sort of looking at the intersection of how we can sort of use this natural inspiration to inform biological engineering.
So the first project we're going to talk about is called Kinematics, and it's a project where we're trying to fuse fashion, software, and 3D printing to examine new ways of producing clothing. How can we sort of take these types of tools and use them not to create just new things that look really cool, but to change the way that we actually create and buy clothing?
So we can think of 3D printing as sort of a new way to construct textiles, which can be a little bit strange because 3D printers make usually these kind of monolithic solid objects, but really sort of textiles are these traditionally human constructed materials. You know, their arrangement in space is really what gives them their properties. It's not whether it's a wool or cotton, but more if it's knit or woven that change its behavior. And so 3D printing offers the potential for much more complex arrangements of matter in space. And what can that allow?
Computation also has a really long history in textiles, which I'm sure you all know about, but it's really cool, so I'll tell you again. The Jacquard Loom is sort of the first machine to use punch cards, and it used them to create these richly illustrated fabrics. So some people like to think about it as maybe the first computer, or at least an important predecessor to computers. So in textiles then are this really cool thing, which are these man-made materials, like metamaterials, like we all like to talk about today, but also sort of historically computationally mediated. So we just started playing with different ideas about how you could make textiles with a 3D printer. And we started with the simplest first idea that we had, which is, well, 3D printers make rigid materials, so what if we just connect together a bunch of rigid triangular pieces with hinges? What would that be like? So we had a little small 3D printer in our studio, a MakerBot, and we used that to 3D print some bracelets and necklaces, which were interconnected by these hinges. And they kind of had a really cool behavior. All the ideas seemed really dumb at first. We found that the resultant objects had a sort of pleasing like hybrid behavior. They're sort of rigid and they're sort of soft. And I immediately sort of started fantasizing about, could I make a dress out of this material? And what would that be like? What would it sound like? How would it feel to wear? But we immediately hit upon a problem. We were making things about the size of a bracelet because our 3D printer is about the size of a bracelet. How could we approach making a large garment? A lot of projects in digital fabrication approach the idea of making a large thing digitally by dividing up into lots of small unique blocks. So you imagine you want to make a building, you divide it into like 1,000 one-of-a-kind unique bricks, which you then need to manually organize and assemble into that amazing thing you designed. So you sort of have ended up taking computation and making your life harder by going from all identical bricks to unique bricks. We had an idea that we could take advantage of the fact that the things we're making are flexible, and what if we can just find a smaller configuration to make them in? Then we could make a garment that could be printed all in one piece where we wouldn't have to cut, sew, or assemble anything. So if you start to think about making a garment all in one piece, you start to think about it a little bit differently. Bodies are three-dimensional, but you're normally thinking about making them out of flat material with flat patterns, which you sort of painstakingly assemble into a garment. We started thinking about making a garment all in one go. So then we were thinking about, can we go from 3D scan of a person to a customized garment that a person can design in a web application to then something that we could print out all in one piece. Whew, it's a lot of words.
But we sort of had this idea, and it took us about a year to go from idea to actually being able to print a customized dress, because there are all these sort of different facets that we had to figure out and kind of debug along the way.
So at the base level, our garment is basically just a bunch of hinges. So even though we ultimately wanted to create a garment or a collection of garments, what we spent our initial time doing is just designing and physically testing many, many, many different types of hinges.
So there are lots of sort of different constraints, different printing methods sort of lend themselves to different designs. We want to miniaturize this part as much as possible while still making it robust, which allows us to make our garments as detailed as we can. And you run into all of these sort of gotchas where printing in different orientations results in a different hinge. And so you print something once, you think it works, you print it again, and all of a sudden you run into problems. So we started prototyping small. And we made this initial necklace, which was the first thing we ever printed, sort of crumpled up. And to our surprise, it actually worked and unfolded. So we scaled up a little bit. And then this is the first piece that was based off of a 3D scan to test fit, scale up a bit more. This was just made off a very quick and dirty Connect scan of Jessica. And that also worked and fit sort of as we expected. So we scaled up again. This time we ran into more problems where we had to sort of redo a bunch of things. This is sort of the first test at scale with more than 1,000 pieces. But we did get this to print as well. So this was very reassuring as we sort of went through our prototyping phases.
All of the things that Jesse just showed you were made in software that we had created that had no user interface whatsoever. So they're very difficult to define the shape and the pattern of the garments, but our goal was ultimately to create something where anyone would be able to customize a garment. So we, at the same time, started working on this software, which is the application you can play with on our website called Kinematics Cloth. This is sort of designed to be an intuitive interface that lets you design garments in 3D. So you start from either a set of body measurements or a 3D scan, and then you can customize the silhouette, shape, pattern, sort of density of structure of the fabric, and then also sort of add on different modules to create different patterns of perforation and solid. And you can, of course, save and share and edit later all of your designs.
So we also had to deal with this idea of 3D scanning and fit. So there's sort of this notion of, well, these 3D scanners are proliferating, you have depth cameras on your phone, you could get 3D scans at home. But the reality is a lot more complicated, and in addition to 3D scanning being very error prone, you also have these sort of semantic issues of you have to be able to identify different parts of the body. Where are the hips? Where are the arms? To be able to design off of that. So we worked with a company called Body Labs, which sort of dealt with that exact issue. They were acquired by Amazon, so they sort of don't exist anymore. But they created a parametric body model based off machine learning that allows you to take a scan or measurements and then get a model that's posable where you can map any point on one person's body to a point on another person's body.
The sort of cornerstone of the project, though, is the simulation chunk. Can we simulate the behavior of these sort of complicated structures in order to reduce their size for printing? To simulate these stresses, we ended up just using a rigid body physics solver. While we initially conceived of just crumpling the garments up to fit them into the printer, it turns out that that's really not the most efficient way to put clothing like in your suitcase or dresser or even a 3D printer build volume. Really the smartest approach that we found was just folding the garments. So that's what we ended up implementing, which reduces the size by over 85%, making it so we can print the dresses in one piece and also more affordably.
We can also use the same simulation to better understand the behavior of the final printed garment because we're making these textiles that have these very strange hybrid soft properties. They vary through space. We don't have a very good intuition for how they're going to behave, so we want to be able to understand that better before we spend a lot of time and money printing one.
So this video shows the first dress that we made and how it was printed. It was printed in Shapeways' Long Island City 3D printing factory using selective laser sintering and nylon. This was a little bit of a scary day for us because we'd been working on this project for about a year, but we'd actually never made a dress and we didn't actually know if it would work. Lots of things seemed like a good idea in the computer, but when you try to do them in real life, sometimes you have massive failure. So about this point, we're sort of wondering, like, is it going to unfold? Is it just a solid lump of plastic? It's made by heating up nylon powder with a laser. So inside of every hinge on the garment, of which there are like over 3,000, there's just dry nylon powder, hopefully, that can be shook out, and then you'll be able to move those hinges. But at this early stage, there's all very tight, and we didn't know. It's also the complicating factor that we had met this curator from the MoMA, Paula Antonelli, and she was interested in maybe acquiring the dress for the Museum of Modern Art collection. She didn't know that we'd never made a dress and that it was just like a conceptual project, and we needed it like literally the next day after this video to give to her for potential acquisition. Luckily, all that powder did shake out and we were able to pull this dress out of the machine, call my friend down, and she tried it on. It was really quite relieving at that point. So this is the first dress that we made. It's a custom fit garment that's an intricately patterned structure of more than 3,000 unique pieces connected by more than 3,000 hinges, all of which was 3D printed in a single piece. While every component is totally rigid, and I have a piece up here you can play with, in aggregate they sort of have this fluid behavior, which allows it to flow and move in response to the body. Unlike traditional fabric, it's very non-uniform. It varies in rigidity, straight, porosity, and pattern through space. But the thing we're most interested in really is that the whole thing is customizable. So you can go from a 3D scan and your own personal style and input to a garment that's really made truly for you. We've done a lot of further explorations on this, including, I'll let Jesse talk.
Yeah, we've probably made about a dozen dresses at this point and sort of done these further explorations. Here, we're coating the thing in a layer of scales or petals, which are also fully customizable in direction and size, which sort of gives sort of more design space for the user. Here's like a quick little video. And then we've also explored other structures like chainmail. The advantage of chainmail is that it's a lot less constrained than these hinges, which allows us to compress it a lot more for printing. So this was a quick project we did for the Formlabs Fuse printer, which has a much smaller build volume. Here we just sort of funnel it into a box, no folding required. And this is sort of the final object, still very much in the prototype phase. The next project we're going to talk about is a collaboration with New Balance, looking at creating 3D printed performance footwear. And this is also sort of exploring the idea of customization, but in an avenue that we haven't really done before, which is focusing more on data and being able to customize shoes to a particular person's performance. So this example looks at pressure data from a midsole, and then we did some initial explorations of different ways to express that data. Some of them are more aesthetic, some of them are more functional.
The approach that we ended up going with for the initial shoes that were released was very inspired by sort of the way that bones have more material and structure in areas where there's more load. So we took that pressure data and translated into a sort of variable density anisotropic Voronoi system. So this is generated using a capacity constrained centroidal Voronoi diagram in 3D. For each of these shoes, they could be customized to the data collected from a specific person. So the one on the left, for instance, is customized to a person who lands on their forefoot when they're running. And the one on the right is customized to somebody who lands on their heel. So they need sort of additional mass and material in that area.
And then we also want to sort of further that customization to allow people to design their own midsole that not only includes like strictly data, but also running preferences and styles where you can sort of sculpt different elements for different types of runners.
So this is an ongoing collaboration. I think the shoes that we showed first were actually released in 2016, but we're still working with New Balance, and actually I think their designers are here today, Chris and Owner and Matt, so if you want to hear about that, you should talk to them. Yeah, cool project. Okay, puzzles.
So this next project is about jigsaw puzzles and sort of reinterpreting traditional craft using computation and digital fabrication. So this is sort of how most people know jigsaw puzzles. They're these cardboard die-cut puzzles. They're still really fun, but they're very repetitive. The shapes are very simple. But this is sort of the history behind jigsaw puzzles before they were mass-produced, is that they were these hand-cut wooden objects that were sort of very artistic. Each cutter had their own style. Every puzzle was different because they were cut freehand. You had much more intricate shapes. Artists would include figural pieces that were thematic in a puzzle. And a lot of that artistry was lost when we switched to mass production. You have these limitations of die manufacturing. And so we were interested in how we could create puzzles inspired by these traditional hand-cut puzzles, but not trying to copy them, but sort of in our own style.
So we started to look at these ammonite fossils. Ammonites are extinct cephalopods that roamed the oceans like over 150 million years ago. And they happen to have these really interesting patterns on their shell called suture patterns. They're actually the walls in between the different chambers of the shell that they would build as they progressively grew larger. These have these really intricate sort of dendritic branching patterns to them. And we thought, wow, that would make a really cool jigsaw puzzle piece. But unfortunately, since they've been extinct for hundreds of millions of years, nobody knows why they formed like that or how. So we sort of hit a dead end with the literature search of researching how you would generate those.
But to us, they seem very reminiscent of a lot of these branching type patterns that sort of all fall under this umbrella of Laplacian growth that has this sort of similar underlying mathematical structure. So we looked at sort of different systems that had this type of branching. One of them is this experiment called the Hellish Oo Cell, where you're injecting a low viscosity fluid into a high viscosity fluid, sort of separated by a very thin layer.
If you haven't done this before, I encourage you all to like go home and get 2 pieces of glass or acrylic and just try putting some paint in between them and separating them, and you'll get these patterns forming as the air sort of rushes in at the side of the plates. It's amazing and so easy to do.
But this is somewhat computationally difficult to simulate, and as designers, we generally want to work with things that are fairly fast and interactive to be able to iterate. So we looked at other systems which are similar, but for various reasons, computationally simpler. So one of those is called dendritic solidification, and it's a crystal growth process that can be modeled using phase field modeling that happens in supercooled fluids. So our general puzzle system layout is we start these certain seeds, they form a general P shape, and then we grow the boundaries to interlock. There are some sort of strange technical details where this phase field simulation is originally designed for solid crystal growing into a liquid environment. We don't have this sort of solid-liquid dichotomy. We have this field of different pieces. So we have to translate this to a multi-phase model, which is physically unrealistic, but we can construct a mathematical model that kind of makes sense.
So using this system, we created our first collection of puzzles, which was called the Radial puzzle. It features, every puzzle has a different artwork on it that was created by Jonathan McCabe. And additionally, every puzzle has a different set of pieces which are grown in the simulation. Each one also has these figural pieces embedded in it, which are inspired by different microscopic organisms you might find by looking under a microscope.
So the idea is sort of just like traditional hand cut puzzles, they're all one of a kind. And then they're laser cut in our studio.
Following this collection of puzzles, we've done a lot more work with puzzles, including developing new different styles of puzzle cuts. This is one of our more recent ones called the Geode Puzzle. They're inspired by, as you can tell, the banded forms of agate. Each puzzle has a different image that's sort of grown by just offsetting lines in a noisy fashion, essentially, and then growing back inwards. They also have this new puzzle cut that we developed, which we call Maze, which you'll see growing in a moment. The maze cut style is sort of grown in a simulation of elastic rods, where each rod lengthens, and as they lengthen, they buckle and begin to sort of intertwine and spiral together in this strange way to make a series of very spindly interlocking pieces.
So one thing we're really interested in is what can you make with computational techniques and digital fabrication that's really hard to do by hand. So by hand, you essentially have to physically cut apart any pieces you want to interlock, because otherwise the joint is just not going to be very clean. But digitally, we can reproduce things very precisely. So in this example, we wanted to make what we call our challenge puzzle, which has no image, has no straight boundary, and in addition, it has all of the purple pieces pull out and form their own sub-puzzle. So what we did is we grew the sub-puzzle first, scattered those pieces inside the larger puzzle, and then grew the larger puzzle around those pieces.
So this puzzle is a challenge puzzle. It's designed to be extra hard. However, we sort of found that you could, even though there was no edge, you could kind of tell where the edge was. The edge pieces had this very defined, frilly look to them. So we started thinking about, well, could you make a puzzle that truly has no edge? Like, what if you just made it so pieces from the left side connect to the right side and pieces from the top connect to the bottom? Then you have a puzzle that doesn't have an edge. Truly, it doesn't make a defined shape, and every time you do it, you get a different shape. So try to like take away what you do when you start a puzzle. You start at the edge. Now there's no edge whatsoever.
So we can kind of think about this in terms of topology. So if we have this sort of tiling topology, that's the same topology as a torus. But inspired by that, we can start to think about different topologies. So what about the topology of a Klein Model? Klein Model is the same as a torus, except that one side flips over. So just like a Klein Model has no orientation, if we make a puzzle like this, we have a puzzle that has no upside and no downside. And so pieces from one side flip over and connect to the other. So we created this puzzle that we called the Infinite Galaxy Puzzle. It has an image from the Hubble telescope, and it's double-sided, so you can never actually see the entire image at once. Every time you assemble it, you see different parts of the galaxy. and just like the other puzzle, has no defined shape. So we're sort of interested in seeing how these sort of new techniques can push traditional crafts in new directions. Not simply, make them cheaper or automate them, but sort of discover new creative outlets.
One of our most recent projects, that we actually have never spoken about before, is a collaboration with Jordan Miller's lab at Rice University, which is focused on developing 3D printed organs. All of our projects, as you've seen, are very inspired by nature, but this is the first time that we've been able to sort of take the algorithms we've just developed inspired by nature and put them back into actually developing things that are alive. So that is very exciting for us.
So this project actually starts from one of our earliest projects from 2010, which is inspired by how veins form in leaves. And there are lots of competing theories of leaf veination. The one that first got our interest is the one on the left, which sort of suggested that there was a relationship between elastic stresses in a growing leaf and the differentiation of veins. So it's sort of very compelling that there's sort of this very simple physical process that leads to the development of complex biological tissue. But the leading theory is the one in the middle called auxin flux canalization. And the idea of that is basically there's a hormone auxin, and where it flows in the growing leaf, it is more likely to flow in the future. And so it's kind of like digging a channel where it is more likely to flow where it's flown before, and almost like a river delta makes these branching channels. And the work that we've done is based on this model proposed by Adam Runyons, which is inspired by that, but not necessarily the most scientifically accurate. It uses this discrete field of hormone sources, and then those hormone sources flow to the closest vein or a neighborhood of close veins. And then each vein grows to the average of sources flowing to it. And as this process repeats, branches form, and then through neighborhoods, closed cells form.
So we sort of read Adam Runyon's paper and then wrote an implementation of what he described in it, and then really started playing with it and exploring how to create new types of sort of pattern spaces, how to play with it in sort of unreal, unphysical ways that don't occur in nature but might be useful for design, such as creating variable density fields or structures that have multiple root points, which is not something you see in leaves. We turned these initial experiments many years ago into a collection of one-of-a-kind jewelry called xylem. These are photochemically etched stainless steel necklaces. Every necklace has a unique leaf shape, and then we grow a unique pattern of veins inside of that. And then people go on our website and they pick the necklace they like and we ship it to them.
So our intention in studying these natural patterns isn't to directly mimic or copy them, but instead to kind of adapt and study their logic into design tools. And so then we can start to ask questions like, okay, what happens when we do this in 3D?
A lot of what we do in our studio is this weird research, but then we also try to turn that into products that can just be everyday products that people could buy. So one of the things that we've done is create a collection of lamps called the Hyphy Lamp, sort of like leaves on a tree. Every lamp is unique, so they all have a different pattern of veins. Some of them all have unique shapes. These are 3D printed using selective laser sintering and nylon.
So we also did kind of these more free-form experiments of 3D growth, but they didn't really have any sort of defined function that we could think of. We weren't really totally sure what to do with them. We made these kind of sculptural pieces that we had in an exhibition called Growing Objects, and we sort of didn't think about them for many years.
And then my slide isn't here. Well, about 3 years ago, we got a weird email from this guy, Jordan Miller, at Rice University. And we didn't know about Jordan, had never met him, but he just sent us an email. They said, like, are you interested in turning your sculptures and the software you created to make them into a way to generate vascular structures for 3D printed organs which we get a lot of weird emails in our studio, and you almost never know whether they're like from real people or not. But we decided to follow up on this email, and it has led to this sort of long collaboration for the past 3 years of working on taking some code that we had already created and turning it into a way to produce 3D printed tissues. There it is. Yep, put in the wrong spot.
So what their lab has done, well, they've worked on many projects, but what sort of started this is they developed a method to do stereolithography for living cells. So they use DLP printing with a hydrogel, which has living cells in suspension. And unlike a lot of other sort of 3D printed tissue projects, you're not building a scaffold, which you then have to put cells in later, and that sort of limits complexity. Everything is printed in place. So this allows us to create really complex forms that wouldn't otherwise be possible.
One of the issues, though, in your 3D printing tissues is that you need to be able to provide nutrients and oxygen to them. If you're printing a thin layer of cells, that could just happen by diffusion. But if you're printing a large block or you intend to print something like an organ, you need to also print vascular structures that can support those tissues. So why he was contacting us is he wanted some help or collaboration with generating the complex vascular structures that you need to support the tissues that they were printing.
So we started just, kind of doing the easiest thing that we could, take the existing system that we already have and grow structures sort of in a variety of densities and details that are printable by his process and see how they turn out. And so the first question is the very basic question of can you print these things? Because these structures are actually a lot more complicated than the test structures that they had done before. And one sort of detail is we're printing sort of the negative of these vascular structures. So we subtract out the structure that we've grown from some shape, and then that shape is printed in hydrogel. So another detail of this is that the structures we're printing need a fluid inlet and a fluid outlet in order to have flow. So in this sort of the simplest scenario is we have two roots, they grow towards each other and merge, and then that's our structure. We could actually grow much more complex things than they're currently able to print, which is sort of this constant push and pull of we would design sort of a thing, and then it would be many, many months before they were actually able to test that object.
But this is sort of part of the attraction of having a generative or procedural model for generating veins, because they're constantly sort of changing what they need. So one week they can print a small thing, the next week they need to be able to print a larger structure. They may need to customize the topology of the topography, whatever, the shape of the veins for different specific bounding volumes, or ultimately to customize to specific people. So we need a system that can adapt and generate vascular structures to any arbitrary sort of bounding shape and density. So although we can generate things like that, this is what they can print at the point where we started working with them, quite a bit less complicated. And they printed some of these things and perfused them with fluid, and we found that they didn't actually work that well. As you can tell, there are areas that, due to the sort of tortuosity of the vein geometry, don't perfuse with fluid. So this is not good.
So we started to look at sort of other modes of growth within the system that we'd explored before to see if we could make something that was more conducive to fluid flow. We wanted to explore the sort of open mode type growth, which is the second one on the screen. But the problem with that is that it doesn't form closed cells. And so you can't simply grow two trees and have them merge. So what we did is we created a sort of modified version of the growth algorithm that we called mutual tree attraction, where in addition to these hormone sources, the growing tips of the veins also flow towards the growing tips of other trees. And then as they get closer and closer, the influence on each other gets stronger and stronger until they merge. Like
these 3D examples. So for a while, the first year or so we were working with Jordan's lab, we were focused on creating vascular structures for just supporting tissues that provided blood. But if you look at a lot of our organs, they actually contain multiple vascular networks, which work in concert and are entangled with one another to provide multiple types of things. So in our lung, for instance, we have an entire network of airways, and those are entangled by a second separate network that it doesn't join that carries blood. So Jordan set us this challenge of, can we come up with a way to generate structures that have multiple interpenetrating networks?
So a really simple example might be a Voronoi diagram and its dual. Probably not the most efficient for fluid flow. And then we also experimented with making these entangled vein-like structures.
Then we did the smart thing, and we worked with Jordan to actually understand how lungs work and how lungs develop in biology, and then tried to design a system that works very similar to that.
So this approach was sort of much more like just purely mimetic than a lot of our other work. So when you look at alveoli structures, you sort of have this grape-like air sac, and then that's sort of ensheathed in this fine mesh of capillaries. So we sort of just said, okay, we have this sort of capillary network which is much more cellular than these hierarchical vein networks that we were working with in leaves. So let's just use a centroidal Voronoi diagram as an approximation of this and try that out. This sort of flows okay, but some of the cross-links don't perfuse very well. So to address that, we sort of made an anisotropic Voronoi where we're elongating in the direction of flow, which makes the branch angles smaller and also makes these cross-link sections smaller.
And then we don't want to do that in 2D, we actually want to do that on the surface of an air sac. So we Boolean together a bunch of spheres to make something approximating an air sac, and then we...
To design our veins, we sort of define a direction of flow by solving the heat equation on our air sac, where the inlet is a source and the outlet is a sink, and we take the gradient of that. We then deform our surface by shrinking in the direction of flow. We generate a centroidal Voronoi diagram on that surface, and then we map that back to the undeformed surface to get our anisotropic cellular net.
So this allows us to create these air sacs which have a directional field of capillary-like blood vessels on them. It's connected to an inlet on one side that carries deoxygenated blood, and then an outlet on the other side which is going to carry the air-filled blood that gets filled due to the proximity of the vessels to the air sac, which is being inflated and deflated. You'll notice that we sort of put all of our tubes at the top. This is because we're trying to create a sort of modular system that allows us to generate things and plug in a bunch of these alveolar units in space. You imagine you take a volume and want to be a lung, you pack in many of these units, and then you could have something that has quite a lot of oxygenation capacity.
So we want to connect this into a hierarchical network, but we actually don't have one network. We have an airway, we have
Oxygenated blood network
and a deoxygenated blood network, like veins and arteries. And so we sort of have to create this way of generating these three intertwined networks. So when we're doing that, we grow the first network, and then we parallel transport a frame field along this network that is constrained to be normal at all of these branch points, and then offset along that frame field. And then at the end of each branch, we instantiate an air sac.
So this allows us to create a system where we can generate these lung-like structures sort of in arbitrary volumes. They could be more two-dimensional or more three-dimensional, depending on what they need to print or test at that point.
So what's been really exciting about this project for us is that it's really been a truly collaborative effort. So as like an art, design, and science collaboration, we're not just brought in to make an image, or we're not just brought in to solve a specific problem, but it's a constant interplay between sort of us pushing them to create more and more complex structures, them pushing us to come up with new ideas and techniques, and discussing, what is possible with this technology.
Our sort of ultimate goal for this project is just to continue working on developing this software that ultimately could be an open source tool for people to use in tissue engineering, which I think is a space where they have need for a lot of complex geometry. And just so you can see how big this thing is, that's how big the thing we're talking about is, which is actually about much bigger than a real alveoli. A real alveoli would fit inside of one of these veins, which is 300 microns. So we're still working at a much larger scale than nature. And if you want to read more about this research, it just was published in Science last month, and this is the paper title.
I think they also published all of the models from this paper, so if you want to print your own vascular network.
They open source the printer and also all of the geometry, so you can download all of that as a supplemental resource. And I think that is our last slide, which is good because we went slightly long. Thank you.